Hi, my name is Akash Babanam, and this is my science fair project, Small Molecules for the Treatment of Bladder Cancer. So bladder cancer is a very deadly cancer that caused 17,770 deaths in last year in 2018. And there are two different types of bladder cancer. One type is muscle invasive bladder cancer, which is almost untreatable and very dangerous, and the second is non-muscle invasive bladder cancer, which stays inside the bladder and does not attack other muscles, but it will develop into muscle invasive bladder cancer if left untreated. So non-muscle invasive bladder cancer is the one that I'm trying to treat today in this experiment. So last year, I attempted to treat bladder cancer with a molecule called urolithin A, which is a compound derived from pomegranates. And what I discovered was that this urolithin A, or UroA, it did inhibit or kill the bladder cancer cells. However, the concentration at which it did so is considered high in clinical use. So what I wanted to do in this year for this experiment was to find a molecule or a compound that was able to inhibit the bladder cancer cells at a lesser concentration. So the compounds that we used were structurally derived from urolithin A, and there are 5 of them. The 5 are ASR 600, OME, UroA, ASR 610, ASR 601, and ASR 602. So these compounds were derived from UroA with some structural modifications. So the urolithin A, as you can see here, so these are the 2 places where the urolithin A was structurally modified, as this is a urolithin A molecule. So in order to test the efficacy of the 6 compounds, not including urolithin A, on the bladder cancer cells, and the bladder cancer cell line that I used was TCC-SUP, which is a bladder cancer cell line. So I use a MTT assay, which is an assay to determine how many of the cells that how many of the cells you have are alive after treatment of any drug or molecule or anything like that. So what I discovered was that the IC50, or the cells, or the number of cells, or the concentration at which the number of cells are halved, so there are 50% of the cells alive. So that number concentration for urolithin A was 15 micromolars. However, for all the other analogs, this concentration was lower than that, and that just shows how these 5 analogs were able to decrease the concentration required to inhibit the bladder cancer cells. So the lowest concentration of IC50, which is again the concentration at which 50% of the cells are dead, was the ASR 600, which had an IC50 of 3.12. The other 4 analogs all had IC50s of either 6.25 or 12.2. So I wanted to use this ASR 600 in my future experiments as I had the lowest concentration and was able to inhibit the bladder cancer cells with the most efficiency. So the first thing that I did was I wanted to see whether or not the cell death caused by the ASR 600 was permanent or temporary. So the way to do this is with a flow cytometry assay, and basically what that does, it determines whether or not the cells are already dead when you treat it with the ASR 600, or if they're currently going through cell death, which is also called apoptosis. So as you can see, if after 24 hours, 25% of the cells were already dead or undergoing cell death, and that means they will die within a couple of hours. And that's very good, as in the MTT assay was done in a period of 72 hours, so in 72 hours it took 3.12 micromolars to inhibit 50% of the cells. But when you did the flow cytometry in 24 hours, already 25% of the cells are dead, and that is considered very good. So the next thing I wanted to check was how exactly is the ASR 600 inhibiting the bladder cancer cells. So I checked a tumor suppression gene, which is a tumor suppression gene called SFRP4, and what that basically does is if that gene is expressed, it suppresses tumors. So in a normal bladder, which is shown here, there's a lot of SFRP4 gene expression, which is shown in the red. So in a normal bladder specimen, there's a lot of this gene, so that means the tumor cannot be expressed. However, if you look at a bladder cancer specimen, there is almost none of this expression, as you can see no red at all. So using this was all done with an immunohistochemistry, which uses tissues. So these are all done with bladder tissues. So basically, what I determined from here was that if there's SFRP4 in the bladder cancer cell line, then the cancer can be inhibited. The bladder cancer cells can be inhibited. So next, what I wanted to check was whether or not the ASR 600 actually did that, and if that's why when you treated bladder cancer cells with TCCSUP with the ASR 600, the bladder cancer cells were inhibited. So I did this using a Western blot assay, which uses proteins and it shows protein expression in a cell treatment. So this SFRP4, as you can see, it is the tumor expression gene, and in the control, which was the bladder cancer cell, before any treatment, there was almost no expression. You can see almost nothing. But then after 72 hours, there was a much higher expression in this SFRP4, as the thicker lines mean that there was more of an expression. So that just means that if you treated the bladder cancer cells with the ASR 600, it allowed the SFRP4 gene to become re-expressed and join back with the bladder cancer cells with that suppressed the tumors, and that suppressed the tumor. So just to confirm this, I also tested one more cell death gene, which if it's there, it'll kill these cancer cells. It's very similar to tumor suppression gene, and that one was Bax. And as you can see, before any treatment, there was also a little, not very much though, expression, but after the 72 hours, there is a more increased amount of expression. That just shows how the ASR 600 was able to re-add this cell death gene to suppress the tumor. So just another thing I wanted to check was that if the genes that are allowing the cancer cells to survive disappear after treatment of ASR 600. And these three proteins that we checked for cell survival that let the cancer cells survive were PAKT, BCL2, and AKT. And after 72 hours of treatment, all of these proteins showed less and less expression, which just confirms the fact that the ASR 600 is indeed the one that is re-adding cell death genes and decreasing cell survival genes. So this can just be showed in this table, in this image that shows TCCSUP cells with the bladder cancer cells, and as you go through cell death, the PAKT and the BCL2 go down as they are less expressed, and the Bax and the SFRP4 are more expressed, and they show more expression. So in conclusion, the data that I collected did indeed confirm the hypothesis, which was if small molecules that are structurally similar to uracil A are used to treat bladder cancer cells, and the efficacy to kill bladder cancer cells is increased, as all five analogs of urolithin A, which was structurally modified from urolithin A, all had a lower IC50 concentration than urolithin A itself. And also, it was also shown that cell death genes will become more expressed after treatment of ASR 600, and cell survival genes will be less expressed in ASR 600. So what we can do with this in future studies is that this could be used in clinical use, as a 3.125 micromolar concentration is considered low, at least compared to the 15 micromolar IC50 concentration in urolithin A. However, it would be nice to find more molecules and more compounds that would be able to bring that IC50 down to nanomolars, which would be most ideal for clinical use. Thank you.